Hello students, good morning. Welcome you all to a new video of Sunanda's tutorial. So after a long time I am uploading this educational video. So it is the part 3 video of your molecular biology chapter. Previously in my two videos of this molecular biology chapter I have discussed about DNA replication in prokaryotes, then DNA replication in eukaryotes. Today we will discuss about the transcription in prokaryotes. Okay, the enzymes involved, in steps and significance. Okay, so let's start. At first, what is transcription? Transcription is the process where the genetic information is transferred from the DNA to mRNA. So it is the first step of your protein synthesis. In the second step, that mRNA will be translated to form a polypeptide chain. Okay, so in transcription it is the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA. In prokaryotes, transcription occur in three phases known as initiation, then elongation, then finally termination. Initiation is the start of transcription, then elongation, which is the actual polymerization or actual mRNA synthesis process. Then finally the process will be terminated in the process of termination. Okay, let's start. Then the enzymes involved in the process of mRNA synthesis. So in case of prokaryotes, the whole mRNA will be synthesized by a single RNA polymerase enzyme which contains multiple polypeptide subunits. In Escherichia coli, the RNA polymerase has following subunits like 2 alpha, 1 beta dash, 1 omega and 1 sigma subunit, and all these enzymes togetherly constitute holoenzyme. So sigma subunit may dissociate from other subunit after initiation, and the rest of the subunits togetherly called as core enzyme. Core enzyme along with the sigma factor known as holoenzyme. Okay, next. So this is the diagrammatic representation of the whole steps of this mRNA transcription or transcription, which is the first step of your protein synthesis. Okay, it consists of three steps. One is initiation, then elongation, then termination. Okay, so let me tell you one thing. Protein synthesis process occur in two steps. One is transcription, one is translation. This transcription process occur inside the nucleus because DNA is present inside the nucleus, and inside that nucleus new mRNA is formed on the template of DNA. Okay, and this transcription process occur in three steps. One is initiation, where the process actually starts, then elongation, where the polymerization or new mRNA synthesis actually occur, and termination, where finally the process stops. Okay, next. Then initiation. During initiation or starting phase, RNA polymerase recognizes a specific site on DNA upstream from the gene that will be transcribed. Which is upstream, suppose this part will going to transcribe. This part is called as upstream. Okay, and this part is called downstream. Okay, so specific promoter site is present here upstream of this transcription region. The holoenzyme bind to that promoter region, which is about 40 to 60 base pair in size, and then initiate transcription a short distance downstream. Within the promoter lies 6 base pair sequence that are particularly important for promoter function. So using convention of calling the first nucleotide of the transcribed sequence called as +1, and these two promoter element lie at the position -10 and -35, that is about 10 and 35 base pair respectively upstream of where transcription will begin. So that actually that holoenzyme will recognize that -10 and -35 sequence. What is -10 and -35? Suppose this part will going to transcribe, okay, and so this is called as upstream, and 10 base pair in the upstream region is called -10, and 35 base pair in the upstream region is called -35. Okay, so promoter, that part is called as promoter site or initiation site, and holoenzyme will recognize that part only. Okay, so the -10 sequence has the consensus because this element has discovered by Pribno, which is known as Pribno box. Okay, it is an important recognition site that interact with the sigma factor of RNA polymerase, because sigma factor is very important for initiation of mRNA synthesis or transcription. The -35 sequence has the consensus TTGACA, and it is very important for DNA unwinding during transcriptional initiation. So this -35 sequence upstream of the transcription site is required for unwinding of DNA, which is TTGACA. One important sequence in prokaryotic promoter is located 10 bases before the transcription start site, which is -10 and commonly known as TACTA box, because that sequence is like this, TA. So it is called as TACTA box. It is important for initiation. So it is the whole picture and whole scenario, whole diagrammatic representation of promoter region or initiation site. So it is the transcription site. Here it is the transcription start site. 10 base pairs before the transcription site is called as -10 region. 35 base pair before the transcription start site is called -35 region. Here the sigma factor bind with this initiation sequence, and this part is called as promoter region. Promoter region, where the holoenzyme or actually the sigma factor binds, which is very important for transcription to start or start site of transcription. Then elongation. After initiation, the sigma factor is released, because that sigma factor is only required for initiation. So after initiation, the sigma factor dissociated from this transcriptional complex and only leave the core enzyme, which is alpha, beta and omega. Alpha, beta dash and omega, which continues the elongation of RNA's transcript. The core enzyme contains catalytic site for polymerization. The RNA polymerase then synthesizes RNA in 5' to 3' direction using 4 ribonucleotides ATP, CTP, GTP and UTP. Okay, ATP, CTP, GTP and UTP. UTP means here the thiamine is absent because mRNA is synthesized though, so thiamine cannot be attached. In place of thiamine, uracil is attached. So these are the precursors and they help to synthesize new mRNA in 5' to 3' direction. Okay. Next, the complex of RNA polymerase, the DNA template and newly formed mRNA all togetherly called as ternary complex, which constitute of 3 components, and the region of unwinding DNA that is undergoing the transcription is called as transcription bubble. Okay. Then the RNA transcript forms transient DNA and RNA hybrid helix with its template strand, but then peel away from the DNA as transcription proceeds. The DNA is unwound ahead of transcription bubble, and after the transcription has passed, the RNA rewinds. Thus, during elongation, RNA polymerase uses the antisense strand as template and synthesizes a complementary RNA molecule. Suppose it is the DNA double helix. Newly formed mRNA will be formed on this strand, okay, on this strand, and it is called as antisense strand. This strand is called as antisense strand or template strand. Okay, then the RNA produced has the same sequence as non-template strand and called as sense strand or coding strand, except that RNA contains U instead of T. So suppose it is the DNA double helix, and newly formed DNA will form, newly formed mRNA will form on this strand, and this strand is called as template strand or nonsense strand. But this strand is called as coding strand or sense strand because it is completely same as this strand, except here T is present, but as this is newly formed mRNA, in place of T it has U or uracil. Okay. Next, termination. Finally, the termination. After elongation, transcription continues until a termination sequence is reached. The initiation site has TA region, so it is called Tacta box. Likewise, the termination sequence contains GC, so it is called as GC rich region. Okay. So the RNA made from DNA palindrome is itself self-complementary, and so base pairs internally to form hairpin structure rich in GC base pairs followed by 4 or more U residues. However, not all termination sites have this hairpin structure. Those that lack such structure require an additional protein called Rho to help recognize the termination site to stop transcription. So the transcription stop site require a Rho factor for termination. Thus, RNA polymerase encounters a termination signal and seizes the transcription and release the newly formed RNA transcript or mRNA transcript and dissociating from DNA. Okay. Then RNA processing. Actually, in prokaryotes, the RNA transcribed from protein coding gene require no modification prior to translation, because in case of eukaryotes they have exons and introns, and introns has to be removed before translation, but prokaryotes lack those introns, so they produce mature mRNA which is ready for translation after transcription. But in case of eukaryotes, they require little bit modification before translation. Okay, so it is all about your transcription in prokaryotes. I hope you will understand this from my video. I have tried my best to teach you the process of prokaryotic transcription in a easy manner. In my next video, I will discuss about the eukaryotic transcription. So please stay tuned, and if you liked it, please hit the like button and please share with your other friends, and please subscribe to my channel Sunanda's Tutorial to get more important videos like this. Thank you.